Hello, today I will be presenting on how much ATP is generated from one molecule of glucose in aerobic cellular respiration. First, it is important to go over the pathway of the cycles. We first start off with glycolysis, in which glucose is converted to pyruvate. Pyruvate is then converted to acetyl-CoA in the pyruvate dehydrogenase complex. This acetyl-CoA then can enter the citric acid cycle, in which high-energy electron carriers such as NADH and FADH2 are generated. These high-energy electron carriers can then enter the electron transport chain to generate ATP. The net reaction of glycolysis is shown above, but let's talk about the steps which lead to the formation of ATP and NADH. First, it is important to note that in the energy investment phase of glycolysis, there's an input of 2 ATP to form fructose 1,6-bisphosphate before its cleavage. Once cleaved, it produces 2 molecules of glycerolaldehyde 3-phosphate, which then becomes oxidized to 1,3-bisphosphoglycerate. This reaction is catalyzed by glycerolaldehyde 3-phosphate dehydrogenase. And here, NAD is reduced to NADH. Since there are 2 cleaved molecules, we get 2 NADH. Next, 1,3-bisphosphoglycerate is further oxidized to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. In this reaction, ADP is phosphorylated to ATP. And again, since we had 2 cleaved molecules, we would produce 2 ATP from this reaction. Another 2 molecules of ATP is generated when phosphophenylpyruvate is oxidized to pyruvate by enzyme pyruvate kinase, in which ADP is again phosphorylated to ATP. This means in glycolysis we actually produce 4 ATP in the energy generation stages, but because of the 2 ATP required from the energy investment, only a net of 2 ATP is formed, and 2 NADH molecules, which are high-energy electron carriers, are also produced by glycolysis. The 2 pyruvate molecules generated from glycolysis first need to be converted to acetyl-CoA through a series of reactions which occur in the active sites of the 3 enzymes which make up the pyruvate dehydrogenase complex. In the last step, the lipoamide needs to be regenerated, and in order to do this, it is oxidized by FAD, which becomes reduced to FADH2. FADH2 is then oxidized back to NAD, which becomes reduced to NADH. And again, since there were 2 pyruvate, a total of 2 NADH molecules is formed by this process. This reaction occurs in E3 of the complex, which is enzyme dihydrolipoyl dehydrogenase. The 2 acetyl-CoA then enter the citric acid cycle. There are 5 key reactions in which are important to generate ATP or the high-energy electron carriers. They are outlined by the boxes, but I will break them down individually. First, isocitrate is oxidized to alpha-ketoglutarate by enzyme isocitrate dehydrogenase. In this reaction, NAD is reduced to form NADH. Second, alpha-ketoglutarate is then oxidized and decarboxylated to succinyl-CoA by the enzyme complex alpha-ketoglutarate dehydrogenase. Again, in this reaction, NAD is reduced to NADH. In this reaction, from succinyl-CoA to succinate, the thioester of succinyl-CoA is cleaved, and this reaction is coupled with a phosphorylation of ADP to ATP, which forms succinate. This reaction is catalyzed by succinyl-CoA synthase. Succinate is then oxidized to fumarate by the enzyme succinate dehydrogenase, in which FAD is used and becomes reduced to FADH2. With the addition of water, fumarate becomes malate, which is then further oxidized to regenerate oxaloacetate, which is needed to keep the citric acid cycle going. The enzyme malate dehydrogenase is required for this catalysis with the help of NAD, which is reduced to NADH. So here is the net reaction of the citric acid cycle, but note, since 2-acetyl-CoA are produced from the pyruvate dehydrogenase complex, we actually get a total of 6 NADH, 2 FADH2, and 2 ATP from this cycle. So let's quickly summarize the important products from these pathways. From glycolysis, we got 2 ATP and 2 NADH. From the pyruvate dehydrogenase complex, we get 2 NADH. And lastly, from the citric acid cycle, we get 6 NADH, 2 FADH2, and 2 ATP. This brings our totals to 4 ATP, 10 NADH, 2 FADH2 per 1 molecule of glucose. Now, these high-energy electron carriers enter the electron transport chain, which consists of 4 major complexes. NADH enters the chain at complex 1, which is called the NADH-Q oxidoreductase. Here, NADH gets oxidized back to NAD, and this process forces 4 protons from the matrix to the intermembrane space. The electrons from NADH are transported through the complex to coenzyme Q, where they will be further transported through the remainder of the complexes. FADH2 enters the chain at complex 2, which is called succinate Q reductase, which is a direct part of the citric acid cycle. Here, FADH2 is oxidized to FAD, and these electrons are also passed to coenzyme Q, where they will be further transported through the remainder of the complexes. The passing of the electrons to complex 3, which is Q cytochrome C oxidoreductase, forces an additional 4 protons from the matrix into the intermembrane space. The electrons are used to reduce cytochrome C in order to transport the electrons to the final complex. At the final complex 4, which is called cytochrome C oxidase, the previously reduced cytochrome C is oxidized, and the electrons from this process is used to generate oxygen. This process forces 2 protons out of the matrix into the intermembrane space. The reason the process of the electron transport chain is so important is because it generates a high concentration of protons in the intermembrane space of the mitochondria as compared to its matrix. This creates a gradient, and therefore ATP synthase, which is at the very end of the transport chain, uses this and pumps the proton in, which provides it with the ability to phosphorylate ADP to ATP, generating our energy. Note, 4 protons are required to form 1 ATP molecule. So, to summarize, complex 1 pumps out 4 protons, complex 2 none, complex 3 an additional 4 protons, and lastly, complex 4 only 2. Now, NADH enters the first complex, which means 1 NADH molecule is contributing to 10 protons being pumped out into the intermembrane space. And since 4 protons are required to make 1 ATP, it means that 1 NADH molecule generates 2.5 molecules of ATP. FADH2 enters at the second complex, which means 1 FADH2 molecule is contributing to 6 protons being pumped out into the intermembrane space. And again, since 4 protons are required to make 1 ATP, this means that 1 FADH2 molecule generates 1.5 molecules of ATP. Finally, based on all this information, we can now finally calculate the total ADP generated. Here is a summary of how many molecules were produced for reference. Based on this, the ATP generated from NADH would be 10 times 2.5, giving us 25 molecules. The ATP generated from FADH2 would be 2 times 1.5, giving us 3 molecules. And then there's a total of 4 ATP made from glycolysis in the citric acid cycle. Adding these numbers gives us a total of 32 ATP per molecule of glucose. However, since the NADH generated by glycolysis is cytoplasmic, it has a lower yield of only 1.5 molecules of ATP per NADH. Therefore, adjusting for this, our real total actually comes out to 30 net ATP per glucose molecule. Thank you for watching and in advance for your feedback.